So we're currently in biotechnology and we're doing a lab on gel electrophoresis. This is a technique that helps us separate molecules based off their size and charge. The ASEP Biotech class just got these new blue light devices that make it much easier for us to do the labs.
Let's do a little summary of what you're doing today. So as a first-time experience with gel electrophoresis, you are basically looking at how far the colors of different candies travel, and that will depend upon their size, the size of the color molecule, as well as their charge. Traditionally, these units are used for DNA. DNA moves towards a positive charge, so that's also what you're going to see with the candy colors as well.